Hello and welcome to Quarantine Lab. Today we're covering Chapter 25. This is today's lab assistant, Lance. Here we have a couple of eggs that have been soaking in vinegar for the last few days in order to remove the shells. We're going to begin by Lance weighing each egg. So he's going to start with our egg that is destined to go into corn syrup, which is hypertonic.
84.6 grams.
Okay, that egg is 84.6 grams. Make sure you write that in your binder.
This one is 86.1 grams.
86.1 for the egg that is going into hypotonic. Now Lance will pour corn syrup over the egg in the hypertonic container. Corn syrup is a sugar solution, and as you remember, sugar is osmotic, so that makes this hypertonic, just like salt. Now he's putting water in the one marked hypotonic. This is just plain water. It has no solutes in it at all, making it hypotonic. Then we will check back on these eggs in a couple of days, see how they look different and how they weigh. So here is a look at them now. See what they look like, and we will compare again soon. Welcome back to Quarantine Lab. It's been a few days since Lance first set up the eggs, and now we are ready to weigh them again. Go ahead. He's starting with the hypotonic egg. And look at how the egg has changed. You may notice it's much larger than when we started.
97.6.
Okay, 97.6 is the weight of the hypotonic egg. Now he has the hypertonic egg. You may notice it's kind of shriveled up and funky looking. And remember, it was in corn syrup, so he's going to rinse it off now a little bit, get that corn syrup off of it. You can definitely tell this egg is shriveled up.
53.
53. So the hypertonic egg is 53.0. So that takes care of this section of the lab with your eggs. And so the changes, you're going to talk about the size of the eggs and how one was bigger, one was shriveled up. And remember, we're dealing with hypertonic, hypotonic, water moving. Remember that corn syrup is sugary, so it has osmotic pressure. It's going to pull water. This next section is just the hormones, so use your notes, use your book, and you're just writing in which hormone is being described with each statement. Now we'll move on to the next experiment. And you may recognize this from Chapter 1. This was one of the first labs you did back in A&P 1, where you blow bubbles in bromethyl blue to see how long it takes you to turn it from blue to yellow. And remember that carbon dioxide is produced when your cells make ATP, and carbon dioxide reacts with water to produce acid. So we begin with bromethyl blue, and it's a pH indicator. As you blow your carbon dioxide in there, the carbon dioxide is going to react with the water, and it's going to form acid, and that acid is going to turn it from blue to yellow. So at the start, we have here just the bromethyl blue and the water in the cups. So we'll go ahead, add your drop of sodium hydroxide. The sodium hydroxide is a base, so it's making this solution basic. Add your drop now. Use your straw and stir it up. Lance, you can go ahead and do yours. Okay, the sodium hydroxide is a base, so it turns it basic. And then we're ready to use the carbon dioxide from their breath to turn it back to acid and make it yellow again. So we're going to begin with the resting measurement. So they are each resting. sitting here at the table. I'm going to start the timer. I'm going to see how long it takes to turn it from blue to yellow. You guys ready? Go. All right, Trent, 7 seconds. Lance, 8 seconds. Okay, now they're going to go exercise for 2 minutes. That's the next instruction here, exercise for 2 minutes. I'm gonna pause the video while they exercise, and then we'll come back and do the next timer. Here's just a little clip of the exercise. Okay, they are done exercising and returning. Go ahead and start. Okay, Lance, 5 seconds. Trent, 6 seconds. So as you can see, their time did go down after exercising, which means they are blowing out more carbon dioxide now because their cells had to make more energy for the exercise they were doing.
What'd you say?
Then the next part of your lab is pH imbalances, and this is where use your book, use your notes. Go through, define these terms, and then here are examples, and you're going to work through, you're going to classify these examples. So, for example, diabetes mellitus. Without insulin, cells can't use glucose to make ATP, so cells use fat for ATP production instead, and using fat produces keto acids. So people with diabetes mellitus tend to get a lot of keto acids, which would then cause metabolic acidosis. So you're going to classify that one as metabolic acidosis, right over here. And I gave you some hints. So from that list, 3 items will be respiratory acidosis, 5 items will be respiratory alkalosis, 7 items metabolic acidosis, and so on. Then when you're done with that, the next one is the same idea, but with fluid imbalances. So again, use your book, use your notes, and you're gonna go through, I've given you some scenarios here, and you're gonna classify each scenario as volume depletion, which will be 5 items that are volume depletion, volume excess, 2 of them are volume excess, dehydration, or water intoxication, or fluid sequestration. So just to do an example together, vomiting. When you vomit, you're losing both the water and solutes. So that would be a volume depletion. In sweating, you lose more water than salt. So there's more water than salt being lost. So that one is a dehydration. So use the part of your notes where I had the beakers and I explained about when you change water or you change salt. And there's Corona Carl hanging out here in my window, watching us do the lab.